So we've gotten about halfway through with mitosis and its phases, and so just remember where we are. We've got the prophase where the chromosomes condense, the microtubules begin to form the mitotic spindle, and then in prometaphase, that spindle attaches to those chromosomes. The nuclear envelope came apart. Of course, it had to come apart first so that attachment can take place. And then during the metaphase, those two spindles essentially establish the middle of the cell and line those chromosomes up. So that brings us to anaphase. And basically what's going to happen here is that after, you remember when we were talking about the chromosomes lining up on metaphase, some of the fibers are going to have to grow, some of them are going to have to shrink in order to make the chromosome move. In anaphase, what happens is it moves into a different mechanism, and these kinetochore microtubules begin to both shrink. And in fact, what's going to happen here is the shrinking is going to take place back at the centrosome. And so as the centrosome basically pulls these subunits off the back end of the microtubule, it's going to very slowly start to pull away. At the same time, all of this is actually controlled by an enzyme called separase, which we will talk a little bit more later in the context of that control, is going to go through and actually digest all of the cohesin protein. Because remember, the cohesin is what holds your two sister chromatids apart. We're about to have the sister chromatids separate and get pulled to opposite sides. So separase is a very key component of that. This entire process can't happen until separase is ready to go in and actually break up that cohesin protein. All of this is to ensure that we separate properly, and what we mean by that is you want 46 chromosomes to go one direction, 46 chromosomes to go the other direction. And remember, sister chromatids are identical to each other, so one going to each side is essentially a copy mechanism. So here is a drawing of anaphase. So notice how the kinetochore microtubules are pulling towards the centrosome, pulling those chromosomes their direction. These are now called chromosomes. They would have been chromatids before, but now once the centromere has separated, then these become chromosomes in and of their own right. And they're both being dragged forward on either side. And this is definitely something you're going to want to look for in the pictures that we'll be looking at in lab, is these tails kind of drift out behind, almost like the tail of a kite, as the chromosome is dragged to either side. And remember, we were talking about the chromosome getting shorter and shorter. So basically what's going on is we're here at the centrosome, we are pulling the chromosome that direction by coming apart. And in fact, there is some disassembly at the other end as well. So we're disassembling in both directions. And what's happening here is the microtubule pulls the sister chromatid this direction. Okay, so here's what it looks like in an actual cell. So you can see the spindle still pretty well defined, and you can see where the spindles are actually attached to the centromeres, and all of these little chromosome tails are hanging out behind. So it's kind of like you're splitting them and dragging them in opposite directions. I want you to also see at this point, because this will come up later, that the microfilaments are starting to take on a slightly different structure to what they were before. Remember in the previous pictures, here, let's go back and see if I can find one. So this is metaphase here. You definitely see the microfilaments are much more dispersed, and now in anaphase they begin to take on a more robust structure to them, as they basically what's happening here is microfilaments are lining up together in parallel with each other, and what we're about to do is create the mechanism that's going to allow the cell to basically pinch into two cells. So this is evidence that the cytokinesis, the separation of the cell, is actually beginning to form, or the processes are actually beginning to form as the split's going to take place. The other thing I want you to notice here too is you'll notice that those interpolar microtubules don't let go yet. Okay, so even though the kinetochores are pulling, or the kinetochore microtubules are pulling those chromosomes to either side, the microtubules that are stabilizing the cell and keeping the two sides of the mitotic spindle in place are still engaged. Okay, so this will take place. This is probably about midway through the process. These guys will reach almost to the other side, probably in this area right here. These will reach to this side right here. And at that point, we move on into the last phase of mitosis, which is called telophase. So basically, telophase, if you want a definition to it, is a return to the normal nucleus. And the two main things that have to happen during telophase, actually it'd be three things. Two things with regards to the nucleus. So the nuclear envelopes are going to have to form again around each set of chromosomes in the daughter cells. The chromosomes will actually begin to unpack and go back to their normal configuration. And of course, the mitotic spindle will have to come apart as well and reform microtubule cytoskeletons on both sides. So here you see the nuclear envelope began to reform, and you see what you'll see is the cytokinesis is well and truly begun at this point, and can't really see it in the picture here, but these guys will begin to decondense. All right, so here we are looking at it a little more closely in real cells. So you can see the nuclear material is beginning to unpack. So instead of those squiggly lines, it's a little more fuzzy. You can definitely see the interactions between the interpolar microtubules, which are still engaged. The astral have actually grown some as we now push this new nucleus towards the center, and we want to have it in the center so that when these two asters begin to reorganize into the cytoskeleton, so you can see another cell here that's not dividing. Remember that microtubule kind of holds the nucleus towards the center and crisscrosses around it, so that's what's going to form here. And you won't be able to really see in this photo, but obviously the nuclear envelope would be reforming in and around the nuclear material. So basically what we'll end up with is a normal cell that has all the components that we started with. All right, and you can see where the two cells are beginning to split, and this is going to be taking place right along that midline, which is why these fibers are still in place, is making sure that midline stays where it should be, and the two cells will basically contract here until they actually become two new cells. That process is what we call cytokinesis, and even though it began back in the anaphase and is taking place during telophase, it will take a few minutes later to finally finish off once the process is done. So this is what we actually call cleavage. The two cells are being cleaved into, or the one cell is being cleaved into two cells. And this is going to be carried out by those microfilaments that we were looking at. And it's hard to make out in those pictures, largely because we're coming at it from the side. If we were able to kind of rotate this around and look down on it, we would see a ring in that area. And what happens is, just like actin fibers will condense on each other in muscles, of course muscles go straight, but in this case it's a ring, and while the muscle cell or the actin fibers contract, it will squeeze the surfaces of the cell together. And of course, it squeezes, and eventually the energetics are going to be so unstable that eventually the two cells just break apart. At that point, then these two mitotic spindles will create the cytoskeleton. Of course, the microfilaments will make their part of the cytoskeleton as well. What you see in that picture are what are called cleavage furrows, and of course, all of this is only true for animal cells. Plant cells will do things a little bit different because they have the cell wall. So here would be looking on something like I think this is probably a frog egg, and you can see well, it would actually be a frog zygote, because we would have been fertilized by now, and then now it's going from the first cleavage into the two-cell stage. So basically what you have here would be a contractile ring just inside of the plasma membrane that's getting tighter and tighter until it finally pinches off into two cells. Okay, so there you can see those fibers here, and this would be in a cell where we are very far along and very close to actually pinching into the two new cells. The other thing here too is you can now definitely see that the nuclear material has definitely started to unpack and is now taking on that nuclear nice organelle shape inside, as this will now reorganize into the microtubules. Okay? Alright, just tying off with some material we've done before. Every time a cell in an organism like us goes through a division cycle, those cells become less and less likely to enter into a division again. So at some point as we get older, and for us it's usually late teens, early 20s, depending on what rate we're maturing at, the cells in our bodies will eventually exit the cell cycle and enter into what we call that G0. And this is true for normal cells, even if you give them everything they need. So if they have lots of nutrients, if you expose them to growth factors and stuff like that, they should, if normal, they will not divide under those conditions. Okay? Alright, so next part of this chapter is going to be on the regulation of these events. And again, this is probably stuff that you may or may not have heard before, so you're definitely going to want to spend some time going through those a little bit more detail. See you then.